Hello everyone, welcome to lecture 2 of Bioinformatics and Application. Today we will explore foundation of bioinformatics and how it helps us understand gene structure, expression and mutations. As an introduction, we will begin with refresher on central dogma and gene architecture. Then we will move into how genotype relates to phenotypes. We will also cover the types and causes of DNA mutations and their effects on proteins. As you already know, central dogma describes how DNA is transcribed into RNA and then translated into proteins. Codons, which are three-letter RNA sequences, guide this process. The genetic code is nearly universal, which allows us to express genes across species. Eukaryotic genes include coding regions called exons and non-coding regions like introns. Regulatory sequences such as promoters and enhancers control gene expression. Transcription starts at the transcription start site, and translation begins at start codon ATG and then ends at one of the stop codons TAG, TAA or TGA. Not all genes are active in every cell. The human genome has about 25,000 genes, and not all of them are expressed in every cell or every tissue or organ. This has significant functional implications, as different combination of expressed genes leads to different phenotypes like your organs. Also, they make up less than 2% of our DNA. The rest include regulatory elements, non-coding regions that influence the gene's activity. Mutations are changes in the DNA sequences, and these changes also alter the outcome. They can result from replication or repair errors or from external factors like viruses. Common types include point mutations, insertions and deletions, and that are also called as indels and copy number variations. Single nucleotide polymorphisms or SNPs involve a change of a single nucleotide. These can be substitutions, deletions or insertions. Insertions or deletions are hard to distinguish, and therefore we often call them indels. SNPs can have different effects. Nonsense mutation introduce premature stop codon. Missense mutations changes an amino acid. Frame shift mutation disrupt the reading frame. And there are some mutations that don't change proteins but may still affect the gene regulation. Those are often called silent mutations. Copy number variation involves duplicating or deleting DNA segments. For example, this green colored gene is amplified in genome A and B, whereas it is deleted in genome C and D. These changes can alter gene dosage and expression and are often linked to disease and developmental disorders. Then there are two different types of mutations depending on where they occur. Somatic mutations occur in body cells and are not inherited. Germline mutations affect reproductive cells and can be passed to offspring. Understanding this distinction is crucial in fields like cancer genomic and inherited disease research. To recap, we have covered gene structure, expression, and then the types and consequences of mutations. Bioinformatics helps us to analyze these changes and understand their biological significance. Our next session, we will explore DNA sequencing, starting with Sanger sequencing, and learn how to interpret sequencing data. See you then.